Hey everyone, welcome back. In one of our previous videos in this course, we dipped our toes into machine learning. We trained an AI to look at DNA and classify it as traditional or improved. But that was supervised learning. We had to give the computer an answer key to learn from. But what happens in the real world when a brand new viral outbreak happens? You sequence 300 viral genomes, but you have no answer key. You don't know if there is one strain, two strain, or five strains. So today we are going to use unsupervised machine learning. Okay, we are going to hand the computer raw data with absolutely no labels and ask it to find hidden patterns. We will use an algorithm called K means clustering. Okay, and this method will be used to automatically discover the hidden substrains of a virus. Right? So let's get started in VS Code. Right. So first we need some data. In a real scenario, you would calculate the GC content and the CpG dinucleotide frequency for each viral sequence. Viruses often have very distinct GC and CpG signatures based on how they evolve. So instead of loading a massive file, we are going to use a brilliant tool in Scikit-Learn called make blobs to simulate this biological data. Right, so let's get started. First of all, we will import the necessary libraries as PLT. We are also going to use pandas this time. And finally, from sklearn.datasets, we will import make blobs. Right. Now what we will do is we will try to simulate about 300 viral samples. We will secretly make three hidden strains. Okay. So yeah, let's get started on that. This is how we make something. Uh, make blobs. After that, we will choose the number of samples as 300. Next, we will choose centers as 3. This will be the 3 hidden strains. Okay. Then cluster 1.5, and finally we will use a random state for reproducibility. Right. This should work, and after running that, we will put this into a pandas data frame. We will use X, and we will name the columns. What should we name it? We can name it GC content. Make sure not to use spaces in places like this, as it becomes complicated later on. So better to use underscores in place of spaces. Frequency. That should do it. And we will also try to print it. Outbreak data generated, and we can display our data frame head. Let's see it, and here you have it. Notice the underscore in our code. Make blobs usually hands back the answers that you see here, right? Which cluster is which is basically the answer. So by using an underscore, we are literally throwing the answers in the trash. So the computer is now completely blind. Okay. So let's plot this raw data to see what we are dealing with first of all, right? So we will use a simple matplotlib scatter plot. So what we will do is plt dot figure figure size can be 8 and 15 like normal. After that, we will create a scatter plot. We will use data frame, GC content, then data frame. Uh, CPG frequency. Let's choose a color as well. We can use gray. Then we will decide alpha 0.6. I think we have an extra bracket somewhere. Yes, it will close over here, and this bracket is not necessary. After that, plt dot title, and we will call it unknown viral samples or raw data, right? What we can do is we can also use a font size, font weight. It's good to use properties like this because we want our data to be more presentable. Although I can skip these things, but it's better to practice these things as well, right? So yeah, X label will be GC content signature, and then Y label will be CP. I guess we accidentally ran it. Let's finish it once. Y label will be CPG dinucleotide frequency. Okay, and then finally plt dot show. Let's run it, and you just see a cloud of gray dots after running this, right? So as a human, you might squint and guess there are a few groupings, right? As you can see over here in these plots. But drawing the exact boundaries by hand is basically impossible, especially if we had 10 dimensions of data instead of 2. So let's unleash the machine learning here, right? So as I mentioned, you will be using k-means. So k-means works by dropping random center points, which are also called centroids, onto the graph. It measures the distance from every dot to these centers, groups them up, and then moves the centers to the mathematical middle of the group. And it repeats this process until the group stop moving. So instead of just talking about it, we can just see how it is done, right? So we just tell it how many strains we suspect, and it will do the rest for us. Okay, so what we will do is from sklearn dot cluster, we are going to import k means. Yeah, I think that should be the correct spelling. We'll check if an error comes later on. K means is equal to k means. Yeah, I think that was the correct spelling. And clusters, we will say 3, right? Random state 42 as previously, then and init as 10, right? So what this does is this initializes the k-means algorithm, asking it to find 3 strings. Okay. Next step, what we will do is we will fit the model and predict the labels in one step. So what we can call it is predicted strains is equal to k-means dot fit predict, and we will enter our data frame. So the AI analyzes the data and assigns a group number, which is 0, 1 or 2 to every virus basically, right? So now we will add the AI's discoveries back into our pandas table. So data frame predicted strain is equal to predicted strains. Okay, and finally we can print it. Uh, Machine, what should we say? Machine learning is complete. Hidden strains identified, right? And in literally 3 lines of code, okay, I think some error is there. What seems to be the error? We have to use n clusters over here. Yeah. So in just 3 lines of code, the AI will evaluate the 300 viruses calculate their mathematical distances and categorize them. But did it work? We don't know, right? So let's plot the results. So what we will do is we will plot the exact same scatter plot, but this time we will color code the dots using the predicted strain. Okay, uh, which is that column we just created using AI, right? So we will also ask the AI to draw an X exactly where it thinks the mathematical center of each strain is, right? So I think we can copy some of the code from here and we can modify it simply. So what we will do here is figure size will be same, then scatter is equal to PLT dot scatter data frame GC content. Okay, CPG frequency is fine. Then here what we will do is uh, after this we will do C is equal to DF predicted strain, and then C map is equal to, here it is. This is just the color coding, okay, and we can remove this, I guess, and alpha is equal to 0.8. Okay, this scatter plot will be color coded by the AI's predictions. Now what we will do is we will also assign centroids is equal to k means dot cluster centers, and then what we will do is we will plot the centroids as big red x's, right? So plt dot scatter centroids from to 0 and centroids from to 1, then color we can use red this time, and marker we will use as capital X, S is equal to 200, label is equal to strain centroid, right? Now finally we will change our title. What we will put here is what can we do? Viral sub strains identified by K means ML, right? Font size we can leave as 14, font weight as bold. Then X label will be DC content signature as previously, and same for Y label. Let's not forget to plot the legend, and finally plt dot show. And look at that. The AI perfectly separated the gray cloud into three distinct biological clades, which is purple, teal, and yellow. The giant red X shows the mathematical center of each viral family. So if this were real epidemiology data from a hospital, you could now isolate the yellow strain and test it to see if it responds differently to vaccines than the purple strain. So what we did was we just turned chaotic data into actionable biology, and this is called unsupervised machine learning. You did not give the computer any answers, you just gave it raw genomic features and the math found the hidden biology. So algorithms like k-means and principal component analysis are used constantly in bioinformatics to group patient populations, discover cell types in single cell RNA sequencing, and track viral evolutions as well. So the code of this notebook will also be on GitHub, and try changing the n clusters to 4 or 5, okay, and see how the AI forcefully divides the data differently, right? So hit the like button, subscribe, and I will see you in the next video.